Starting out by just thanking Alfonso and Heather and Brooke for inviting us, giving us this opportunity to tell you a little bit about what we are thinking about and what we're trying to do. And what we are here, you see this list of names. This is a group that is unified in their interest in using microscopy as a tool for life detection. And what unifies us in this interest is that we just recently were awarded an award from the NASA Matisse program. So we're still in the honeymoon period and very excited about this and excited to tell you about the approach that we want to look at a little closer. Myself, I'm a microbiologist with interest in astrobiology, and I'm sure there are other microbiologists in the audience. And if not, probably at least people who have endured a microbiology class. And if you have, what you may remember on day one, probably 15 minutes into the first lecture, is some context on how microbiology got started. And it's just about impossible to talk about the foundation of microbiology without talking about the technological advance that made this possible. For those of you who don't know, a Dutch gentleman by the name of Antonie van Leeuwenhoek back in the 17th century was the first to actually visualize the microbes that we refer to now as bacteria and archaea. Microscopes were around. It's not like he invented the microscope, but what he was excellent at as a lens crafter was crafting these single lenses, which allowed him to gain a resolution of about 1.5 micron. It's actually amazing that he was able to visualize cells that were the sizes of bacteria using this system. And over his lifetime, he made about 500 of these microscopes. Microscopy is not just a tool of the early fathers of microbiology. It's still widely used. You may talk to a microbiologist and ask them, oh, do you use a microscope for life detection? And you might stump them. They might say, they might not have an answer, or they might say no. But actually, we use microscopes for life detection in our own laboratories every day. I want to give you an example from my own research where microscopy itself was part of our objective here. And that objective, believe it or not, was life detection here on Earth. So for the past decade, I've been fortunate enough to be involved in some research efforts, which have been the first to drill through the Antarctic ice sheet and sample lakes that are at the base of them. So when we do this, when we did this, especially for the first time, one of the questions was, believe it or not, is there life there? The images that you're seeing here are all micrographs. These that you're seeing in B, C, and D, you're recognizing as scanning electron micrographs, I'm sure. The intimate detail they give on cells and cell structures, which are highlighted by the yellow arrows. We did not have a scanning electron microscope with us in the middle of the West Antarctic ice sheet. But what we did have with us is a fluorescence microscope. This image that you see here, many of you probably recognize as an epifluorescent cell, as an epifluorescent micrograph. And these cells, we are visualizing them because we add a dye that intercalates in their DNA and very conveniently fluoresces when hit with very specific wavelengths of UV light. So this image would be the kinds of images that we would have had in our hand about an hour after this water would have come up with a borehole. And this was an instrumental technique in us determining whether or not this was an ecosystem that actually had life in it. Now, how I came to be thinking a little bit more about the application of this to astrobiology is that I was lucky enough a few years ago to be a member of the science definition team for a mission proposal for a Europa lander study. my sense is that most of you on the call are familiar with this study, and I won't go into a lot of details other than to reiterate that the objectives of this report were to seek life by looking for life, either morphological or textural features of life. And this included objects down to the resolution of microscopy, 200 nanometers. If you are if you're someone who was never really an advocate of microscopy and maybe were outraged by the fact that this was a proposed instrument in this Lander study, you can pretty much, I think, blame myself and Allison Murray, is that we were very strong voices that this was a very important tool that should be considered on life detection missions. Of course, one of the things that was of concern is that while there is some flight heritage for microscopes, a microscope that has been designed specifically for life detection doesn't exist. And one of the recommendations that we made in the report is that NASA actually invest in developing this as a resource that we viewed as would be very important for life detection. And so it's actually been encouraging that there have been funded projects that have advanced this. I just wanted to take a little time to discuss some of the advantages of using microscopy as a method of life detection. We all appreciate the fact that we could encounter a new life form that would be chemically different from life as we know it. But I think it's at least hard for me to imagine that life form not having a body. So while we may not be able to know what it's composed of, we can have an expectation of what it might look like. And so microscopy is one agnostic approach that could allow us to identify an alien life form that we may not know the chemical composition of. There are a lot of good reasons to think that if we go to a place like an ocean world, that the kind of organisms that we're going to see are microbial. One of the things that benefits us is that normally the smaller the organism, the more numerous it is. So that could actually bode well for our ability to detect it. But even if these organisms are very low abundances that we feel could be difficult to detect, it still affords us the opportunity to actually concentrate these cells as a way to enhance the detection. And apart from just taking an image of an extraterrestrial cell, the size and the structures that are features of this that could be very important measures of biogenicity. The size of the cells themselves can give us important information on how easily substrates or products are actually able to enter or exit the cell. And then the final strength that I might say with microscopy is that it can be coupled with approaches that allow us to understand the chemical composition of that. Just seeing something that looks like something we might think is biological is one thing, but understanding a little bit more of what it's composed of is an attractive advantage. This allows us to actually target fluorescent molecules in completely non-invasive approaches. We'll also tell you, though, about approaches that we're investigating to actually stain based on chemical composition. And as all of you are aware, these microscopic techniques can actually be mated with techniques in spectroscopy, which can give us very detailed information on the visualized target. I might just say that the final advantage that I might say is that if this is a mission that is in my lifetime, which I hope it is, and we are going to have the taxpayers spend dozens of billions of dollars to figure out whether or not there is another form of life in our solar system, are we really not going to take a picture of it? I find that hard to accept. And I might sort of have us wonder what the PR value would be of a picture of the first alien life form. We're very familiar with the image that I'm showing here, and the PR value that image has is probably immeasurable, but I believe it would pale in comparison to a picture of the first alien. So this represents my transitional slide, in that to do all these things with this complex task, what would this microscope look like? And we now exit my area of expertise. I can tell you what this microscope won't look like. It will not look like any of the images that I'm showing here. And so with that, I'm going to turn this over to the other half of my team.
That was excellent, Brent. Thanks. We can't hear anybody. I don't know if you guys are saying anything yet.
Yes. Can you hear me now?
We can hear you now.
Great. Well, this is a redundant title slide. So I'm Richard Quinn from NASA Ames Research Center, and we're going to start off here with Nathan telling me how to use the mouse.
We currently see your note slides. If you want to switch it to.
Okay. Okay, so I'll turn it over first to Allison Murray to continue sort of the transition from science to technology.
They can see the notes slide though. We might want to change the view.
Under display settings.
There
we go.
Excellent.
Okay. And I'm Allison Murray. I am part of this team. I'm also one of the co-leads of the Network for Ocean Worlds, and I come from Desert Research Institute in Reno, Nevada. So I guess the foundation for the microscope project that we're going to tell you about was stimulated from some of the Europa Lander science goals and objectives. And so we thought we would just run through these for those who aren't familiar with them. But one of the goals that was identified was searching for evidence of life, was to not only assess habitability, but to look for biosignatures or signs of life. So the objectives here, the first one is identify and characterize morphological textural indicators of life with submicron bright field imaging. Spatial fluorescence are the approaches that we are using. The second objective was to detect and characterize inorganic indicators of life. In the case of our project, we're using native fluorescence and to identify minerals. And the third objective was to detect and characterize any organic indicators of life. The approach that we're using is to detect proteins, polypeptides or amine-containing compounds, lipids that are in organized structures like membranes, and nucleic acids. And we're doing that using fluorescence, either native fluorescence or added stains that fluoresce and bind to these molecules in a specific manner. And then the last objective was to determine the presence of environmental factors essential for life or habitability. And so in this case, having embedded sensors for pH, EH, or conductivity can also provide characteristics of the sample being looked at.
You have to hit the arrow behind the
I can't see it. Sorry.
You want to take this one, or do you?
You can go.
Okay, so what we'll do very quickly here is walk through some of the technology, the implementation that we're looking at to sort of support the goals that Allison just mentioned. The history of this project is, Brent said, the Europa Lander report came out and we applied for Coltech funding and were successful. We followed that with IC2 funding, and now, as Brent mentioned, we're now in our third round of funding with Matisse. The microscope has three operational modes. First is bright field imaging, so we can actually see something, get that picture, and characterize, morphology. We also have the ability to excite fluorescence with deep UV, and so we can look at native luminescence. In particular, we're looking for the presence of aromatic functionalities, proteins, and also to understand some information about the mineralogy. And then finally, as Allison mentioned, we use a combination of fluorescent stains to stain structural biomarkers. And I'll mention behind me here is Pavel Clear, and he's a postdoc, and he's sort of leading the effort now on the microscopy stains. And he's sort of taken over from Kathy Bywaters, who many of you know, who's now at Honeybee, but she had been here at Ames for quite some time and was leading that effort. But now Pavel has taken over. Next slide. So very quickly, I'll walk through the subsystems. So if you look at the CAD model on the right, the microscope, it's a complete system that has the ability to handle samples, ingest samples, and it's in a form factor of 30 centimeters. And the reason for this form factor is it was actually what was dictated by the Europa Lander proposal information package. So before the Europa Lander got, sort of put off to the side with the new decadal, they had issued, we're expecting to issue a call for instruments, and they had gone as far as providing the information package. So we're really worked hard the first two rounds to fit in this volume, and we're continuing along this path. And some people may say, well, why don't you, relax a little bit? Europa is the hardest implementation. And that's why we want to keep pursuing it. We're trying to push this instrument up to a TRL6 or close to a TRL6, and you really have to understand and design to a particular mission implementation to hit that mark. So the subsystems here from top to bottom are the camera board, the optics, or the microscope itself. And in just a few slides, I'm going to hand the talk over to Nathan Bramall, who's been developing the optics subsystem, and he'll give you more details about that. Below that is a microscope objective, a sample filter stage, sample transfer dock, a hermetic valve block. So that's actually so we can hermetically seal our fluids that we use for sample manipulation and staining, a fluidics manifold G, and then finally where we store fluids for our chemistry and also where waste is placed. Next slide. So the sample transfer system we designed specifically to the CADME's sample handling system, and that was also an IC2 project run out of Goddard. And so the key features here is we have a 5 cc sample cup, and this was dictated by the science definition report, and that flowed down into the proposal information package. We can change this up. We can resize that sample cup to whatever mission might impose on us, but right now we are designing to 5 cc's. The Honeybee Robotics, actually the ones that implemented and built this thing, and it's undergone environmental testing, and as a subsystem, would say that this is pretty close to TRL6. The fluidic subsystem. So this is, most of you in the audience here who do instrument development, I'm sure you realize that, a key challenge is sample processing, sample manipulation. And the way we do this is with a sample, a monolithic fluid manifold. And the heritage here is space biology. And this part of the project is really overseen by Tony Rico. And part of what we have in this package are pH sensors, ORP to get oxidation reduction potential, pressure measurements for feedback control, so we can do automated sample manipulation and processing. The fluorescent stains, which we've already mentioned a few times, we have two candidate sets of dyes right now that we have done some radiation testing on. On the lower left there, a primulant stain, which is for lipids, and the red is an irradiated stain. So it's looking at the staining efficiency after irradiation with 300 kilorads, which was the Europa Lander requirement. And then the white is the non-irradiated control. And likewise on the right, acridine orange, some of you may be familiar with, so that's a DNA stain. Next slide. So we have implemented in this a sample carousel, so we can process multiple samples. In this current configuration, we have 3 sets of inline filters. And again, well, why 7? Why this? We're still working to the constraints of the Europa Lander, where in the end, there was a requirement to analyze 3 samples. So this would give us 3 samples, 3 replicates, and a blank. For each of these inline filters, we filter from 10 micron to 1 micron to 0.2 micron, so we can get size sorting to optimize our imaging. The picture on the bottom there is just a detail of how these things look and how they're made. They're actually etched on a silicon wafer with a silicon nitride membrane with the appropriate size holes are ion milled in them. And then the filter geometry here is optimized clearly for the field of view of the objective. You can see the filter stage on the left, the green arrow, the objective above it. Next slide. So as part of the electronic subsystem, we're building a custom camera based on the E2V CIS imaging sensor. And this is been flight qualified to 200 kilorad as part of the JUICE mission. So it's flying as just part of their, not microscopic, but just their imaging system on that spacecraft. The sensor, we map 1.8 microns per pixel. So Nathan will probably talk a little more about this, but really we're at the limit, our resolution limit of light diffraction. So we really can't we're basically as good as we're able to get with optical imaging unless we go to a super resolution mode. Imaging processor software is being ported to a rad-hard microcontroller. There's actually been some newer developments on rad-tolerant microprocessors that we're looking at, so we're actually switching over to a new one. And then the autonomous image processing, where we do Z-stacking, data interpretation, data compression, and I think Nathan will be speaking a little bit about that. And the next slide. And so this is the details of the optics subsystem, and I'll hand it over to Nathan.
Okay. Yeah, so in here is the optical microscope. It's 10 by 10 by 13 centimeters in volume. It comprises like 3 stages, essentially. It has an excitation stage at the bottom, which has 4 LEDs in it that are being combined together, ranging from the deep ultraviolet to the visible at 275, 375, 470, and 525 nanometers. Above that is a filter wheel stage, which blocks out the light that you don't want to see for fluorescence imaging. It also allows for bright field imaging. There are 5 positions on it. And the filters for emission, we currently have at 334, 470, 529, and 579 nanometers to match with the different dye sets that we're working with. At the top, we have some beam folding optics, which wrap the beam around. Normally it'd be about a 200 millimeter focal length, but we wrap it around so it takes up only a few centimeters of height and then focuses onto a camera board above. The components, the key components have been radiation tested at 300 kilораds, and these include representative bandpass filters, both single and multi-band versions, lenses, lens materials, and even some of the LEDs we've already tested, most of the LEDs actually. It's also been tested, assembled at shock and vibe up to the GFST 7000. So it all is really mechanically robust. The exception is that in this photograph is a COTS piezo stage that is being replaced with a custom piezo stage that is designed for shock and vibe tolerance. The piezo stage provides up to 400 microns focusing and movement, and with an accuracy of 1 nanometer. And the entire package is quite functional and small. This is showing how we accommodate for the depth of field of large objects. We take slices of the object and Z-stack it. Shown on the right is the focal plane moving through, or shown on the left is the focal plane moving through a field of sand and debris, small dust particles. And on the right, the stacked image showing how you can take many partially in focus images, stack them together to get one in focus image with a large depth of field. This allows us to image large particles on the larger filters along at high resolution, submicron resolution, and also image in small particles in the smallest sieve. This shows some of the, one of the images of the Bacillus megaterium. This is the autofluorescence imaging of proteins of the bacillus. You can see them quite clearly here. The resolution of the microscope is easily at 2,000 line pairs per millimeter. That is at 400 nanometers, middle of the road. In the deep ultraviolet, it's much better, and in the further visible, it's worse. And that's just the physics. It's proportional to the wavelength. But we are around diffraction limited here. The objective shown here is a 40x objective. That allows us pretty good resolution, as you can see, and with a reasonable depth of field and a working distance, which is quite important since we're viewing microfluidic flow cells. You can't have a 100x oil immersion lens that doesn't have a large working distance. Some of the other things we're doing right now in progress is we're developing another flat field microscope objective. This is already in production for us. It's a 50x 0.3 NA. Works the deep UV invisible. Unlike other deep ultraviolet objectives you may be able to buy, this has a flat field. Their objectives are curvature on them, so they're never entirely in focus in a single plane, but in a hemispherical package. This actually gets really good resolution, above 22,000 line pairs per millimeter, closer to pure diffraction limited 2200. And we're also expanding the microscopy techniques to include multispectral and hyperspectral imaging to support more detailed investigations of both organics and inorganics. These will be designed in similarly rugged packages suitable for flight and automated. Yes, want to close?
Well, that sort of wraps up the technical portion of the talk, and we went through that very quickly because we know that, or hopefully people will have a lot of questions and we can, discuss the details.
That was perfectly timed. That was excellent.